Thank you for stopping by our ASBMB virtual poster session, Accelerating Adoptive Cell Therapy Development Using Sound Waves. I'm Michael, an application scientist here at Lumix. If you have any questions, we'd love to hear from you. My email can be found on the title slide and will be shown again at the end of this session. I'd also like to thank the organizers for their work in setting up this virtual poster session during these extraordinary times. Your effort in doing this is very much appreciated. Before we begin, I'd like to provide a little background, and hopefully, by the end of this presentation, you will agree that avidity measurements are a novel and useful metric for characterizing immunotherapeutics. The immune system is a complex network of specialized organs, tissues, and cells. At the most basic level, the skin and mucosal membranes form a barrier to the outside world. At the cellular level, the intricate crosstalk between innate and adaptive immune cells culminates in pathogen-specific immune reactions that protect the host from disease-causing agents and aberrantly functioning host cells. Direct cell-cell contacts form an integral part of this immune response and are the foundation of important events such as antigen presentation, cytotoxic cell killing, and phagocytosis, as a few examples. Commonly used methods in early immunotherapy development focus on the strength of interactions between single receptors and ligands. This is true in the CAR-T and antibody-based therapeutic fields, as well as in therapies that utilize the T-cell antigen receptor, TCR. Single molecule affinity measurements, however, miss the complexity of cell-cell interactions, which are comprised of multiple receptors of varying affinities. The collective bond strength of these non-covalent interactions is termed avidity and is a better descriptor of the overall cell-cell interaction. By measuring avidity rather than affinity, we propose a new means of accelerating immunotherapeutic development. Enter the z-Movi platform. I would now like to take you through our optimized workflow. The z-Movi cell avidity analyzer uses acoustic waves to measure the interaction strength between effector and target cells. In a typical z-Movi workflow, target cells are cultured as a monolayer on the microfluidic chamber. Once a confluent monolayer has been established, the effector cells can then be flowed into the chip. The effectors and targets are then co-cultured for a defined period of time, typically 10 minutes, to allow for the formation of an immunological synapse. A user-defined force ramp can then be applied to the cell-cell conjugates to measure their interaction strength. This is done by passing an electric current through the piezo element on the surface of the chip, which generates standing waves in the microfluidic chamber. Modulating the voltage changes the amplitude and frequency of the waves, and thus the magnitude of force experienced by the cells. Once the effector cells have detached from their targets, our inbuilt tracking software registers the detachment events and tracks the cells as they are levitated towards the acoustic nodes. The z-Movi uses fluorescent imaging to track cell detachment events. As the magnitude of force is gradually increased during the force ramp, fluorescently labeled cells are tracked in real time with our WAVE software platform. In this example, non-transduced T cells on the left are compared with CAR-transduced T cells on the right, both against antigen-expressing target cells. Without any activation signal, the non-transduced cells rapidly detach from the target cell monolayer, as shown by the black curve on the right. The antigen-specific CAR-T cells detach more gradually from the target layer, as shown by the red avidity curve on the right. This shift in curve between the non-specific and specific T cells represents an increase in the interaction strength between the T cell and the target cell, and a corresponding increase in the amount of force necessary to detach individual cell-cell conjugates. The data provided by the z-Movi thus provides information on cell-cell interactions on a single cell level. I'd now like to go through the data we've obtained through a series of exciting collaborations with UPenn, Lucid Bio, the University of Lausanne, and Glycostem. In a collaboration with the Albelda Lab at UPenn, we investigated CAR-T cell avidity using two previously described cell lines. The CAR-T cell lines will be referred to as high-functioning or low-functioning, based on in vitro cytokine release and cytotoxicity assays. We screened these two cell lines against target HT1080 cells that were transduced as target antigen null on the top left, target antigen low, middle, or target antigen high, lower left. What is immediately apparent is that antigen-driven differences in CAR-T cell avidity, with both cell lines showing increased interaction with the antigen high cells. Both the high function and low functioning CAR-T cells showed no adherence to the antigen null target cell line, as observed by the non-transduced controls. By establishing a cutoff threshold at 180 piconewtons, we can clearly see the differences in binding avidity between the cell lines against cells under each of the antigen expression conditions, shown in the middle panels. Nicely, we find that mean detachment forces, shown in the top right, correspond well with cytotoxicity measurements and interferon gamma release, shown as the middle right and lower right panels. To better compare between experimental groups, we proposed Avidity Score as a more robust comparison tool. In calculating this, we scale the binding avidity of a test group to the control cells. Thus, Avidity Score is the mean rupture force of the test group divided by the mean rupture force of the non-transduced negative control. In calculating the avidity score for each CAR-T cell line relative to the non-transduced controls, we see that avidity corresponds strongly to CAR-T functionality, and that this variable is influenced by antigen expression on the target cells, as is shown in the figure on the right. To further establish the utility of avidity measurements in immunotherapeutic development, we worked with the Mayer Group at King's College London to screen a panel of CAR-T cell clones against an FDA-approved CAR-T construct. We'll start at the top left. It's a busy graph, but I will talk you through it. What you see is a long-term tumor viability assay. On the y-axis is tumor viability as a percentage, and on the x-axis, stimulation cycle in weeks. Each CAR-T cell population was re-stimulated with fresh tumor cells, after which viability was determined. Whenever a tumor grows out, this is an indication that the CAR-T cells are no longer active. If you look at the green and light blue lines, you can see that CAR-2 and CAR-1 perform as well as the original CAR construct. But what is more interesting is that CARs 3, 4, and 5, indicated by the pink, dark blue, and yellow lines, outperform the original CAR. In fact, after 14 weeks of re-stimulation, CAR-5 is still completely able to eradicate the tumor target. Late outgrowth of the tumor suggests that these CAR-T cell constructs are still active, which is very interesting. It would be great if avidity measurements could be an indicator for this delay in exhaustion. On the top right panel, you can see the avidity curves for the same CAR-T cell populations, shown as an average of 10 to 11 individual runs per CAR-T cell curve. What you immediately see is that the original CAR has the highest avidity, and perhaps even more interesting, that the best serial killer shows an intermediate avidity. If we then draw a gate to control for background binding, shown in the middle left panel, the percentage of specifically bound T cells reveals similar results, with the FDA-approved analog CAR standing out. The lower left panel gives us a population view of these CAR constructs. These data indicate that CAR-T cells do not require the highest avidity to be the most efficient serial killer. If we then look at the Avidity Score, which is shown here for one of the 3 donors, you can appreciate that the FDA-approved analog is ranked as the highest binder, with CAR-1 and CAR-4 the lowest. So what is interesting is that these data together suggest a Goldilocks avidity window, which can be seen as an area for selecting candidate CARs with optimal functionality. The avidity score can be used to identify these potential leads. Avidity measurements allow for a quick decision-making, with assays being performed on the order of hours rather than weeks. In the lab of Dr. Nathalie Rufer of the University of Lausanne, their aim is to demonstrate a functional optimum of T cells following affinity maturation of their TCRs. One challenge that they are facing is that standard in vitro assays are laborious and can lead to variable outcomes. Rufer and her team were interested in using the z-Movi to quickly discriminate T cells with affinity-matured TCRs. Here you can see 2 example figures that characterize their affinity-matured TCRs. On the left, you can see results from a cytokine secretion assay, and on the right, results from a cytotoxicity assay. The TCRs are ranked from wild type to very high affinity. The TCRs that they used in their avidity measurements are indicated with arrows. The affinity of each receptor was determined by SPR and compared to the wild type. DM beta had 11 times higher affinity, while QM alpha was 153 times higher. In contrast to the logical ranking in affinity, the functional assays appear as a bell-shaped curve, where DM beta is clearly outperforming the others in both assays. To show that this effect is antigen-specific, NA8 cells without peptide pulsing were not killed at all. Here, TCR affinity does not match T cell functionality, so can avidity be a better predictor? In the 3 leftmost panels, you can see the avidity curves that they obtained. The top is wild type, the middle is DM beta, and the lower is QM alpha. It is important to note that background binding to empty target cells varies amongst TCR transduced cells, showing the importance of taking individual negative controls per tested TCR. This can be seen from the thresholded data in the middle 3 panels. The same can be seen when looking at individual detachment forces of single cells, shown as the right 3 figures. However, the repeat runs on peptide pulse target cells are highly similar. Using the avidity score, where you can correct for the variation in background binding, it is clear that avidity correlates to T cell function. In line with the bell-shaped relation, DM beta appears to be the optimally matured TCR. Thus, z-Movi avidity results match functional assays. Having now shown you data on transgenic T cells, I'd like to move on to an innate immune cell-based therapeutic. In our work with glycostem, we were able to show strong differences in NK cell binding avidity between cells derived from 2 different donors. Each donor is shown here as a different color. The overall detachment force from 2 runs shows that donor 2 had considerably higher binding than donor 1 for the target cells, as seen in the top left and bottom left panels. When we threshold the data as before, as seen in the top right, we can see that the fraction of bound cells corresponds strongly with the killing efficiency on the lower right panel, showing that in this system cell avidity is a direct readout of cytotoxic potential. Finally, as a proof of principle pilot study, we investigated the potential for our platform in the development of T cell dependent bispecific antibodies. Like CAR T cell therapies, bispecific antibodies can be designed to redirect T-cell function towards a specific tumor-associated antigen. Previous work has shown that bispecific antibodies drive T-cell activation in a manner analogous to the endogenous peptide MHC TCR interaction. We found that T-cells treated with a bispecific antibody bound more strongly to their targets than untreated control cells or T-cells treated with a non-specific bispecific antibody, as is shown by the top left and bottom left panels. As before, we see a higher fraction of bound cells in the antigen-specific test group, seen in the top right. When we control for background binding using our Avidity Score measurement, seen on the bottom right, we can see clear differences between the two conditions. These results are interesting and warrant further investigation. With our system, we were able to detect differences in the avidity measurements between the two groups. As avidity takes into account more physiological cell-cell interactions, we present the z-Movi as a novel means for developing new immune cell-based therapies. I would now like to summarize this presentation. In conclusion, we have shown the utility of avidity measurements in assessing CAR-T cell function, noting that we detect antigen expression level differences in avidity score. We have also seen that different CAR-T cell clones show differences in overall binding avidity, and that more intermediate levels of cell-cell interaction may result in improved CAR-T longevity. Together, these findings show that the z-Movi has a role in defining not only CAR-T cell functionality, but in optimizing on-target effects while minimizing off-target cell-cell interactions. We have also shown that with regards to TCR-based therapies, avidity is a better predictor of T-cell functionality, as intermediate affinity TCRs showed not only the highest avidity for their targets, but also the highest degree of cytokine release and cytotoxicity. Thus, understanding avidity in this context allows for the tuning of TCR-based therapies. We have also been able to recapitulate our findings on innate immune cells by showing that enhanced avidity of NK cells for their targets directly relates to enhanced cytotoxicity. Finally, we also present pilot data in the field of T-cell dependent bispecifics, showing that avidity measurements are a valid readout of bispecific antibody-driven cell-cell interactions. Optimization of this parameter may yield to more therapeutically potent immunotherapies. For any questions or comments, or to discuss the possibility of an instrument demo, please feel free to get in touch. My email is m.harris@lumix.com and can be found on the final slide of this presentation. Thank you all for stopping by this virtual poster session. Before we head out, I would like to thank all of our wonderful collaborators at UPenn, Lucid Bio, Glycostem, and the University of Lausanne. Thank you all for stopping by. Given the challenging times that we are all currently facing, we here at Lumix are working hard to support you as best we can. As such, we are currently coordinating with our dedicated team of scientists in Amsterdam to perform z-Movi demos in-house. If you have an experiment in mind, or just want to learn a little bit more about our avidity analysis technology, we'd love to hear from you. We are happy to provide any additional information or discuss the potential applications for our system. Please feel free to reach out. Thanks again.